Biosimilar, with a product which is similar to that of a original product which is manufactured by some other company. That is called a biosimilar. Patent. If you have a creative or innovative idea, or you have created an innovative product, in order to prevent some other or people copying it, you can get it patented so that no one else can copy your innovative idea or innovative product. After the period of the patency only, someone can copy your innovative idea or innovative product. So, biosimilars are the compounds which are similar to that of the original compound, innovative compound, that can be prepared after the patent is expired. And this biosimilar is not equivalent to the original compound. It is just similar to that of the original compound. Biosimilars are also known as follow-on biologics. So this biosimilars are the copy of the product which is already been marketed. So what is the reason for preparing this biosimilars is, when you prepare a biosimilar, that can be available at less cost. But the procedure for preparing the original compound is more complex, and no one can prepare the original compound or innovative compound as the original one, but they can prepare the similar one to that of the reference. Various agencies or authorities have given a definition for this biosimilars. It is WHO, US FDA, European Medical Agency and Health Canada. Biosimilars indicates bio in living organisms. So the source for the biosimilars preparation is from the living organisms, whereas generic one is prepared from the chemical synthesis. And the size will be larger for the biosimilar, small for the generic. And the structure is well defined for the generic. It is complex for this biosimilars. And the generic one are more stable when compared to the biosimilars. And generic one, that doesn't produce any immunological reactions, whereas the biosimilars produce immunological reactions. The bioequivalence with the reference product, the original product, will be similar to that of the generic, and it is not bioequivalent for the biosimilars. And the cost will be high for the biosimilars. For the generic one, it will be low. What are the principles for the development of biosimilars? So when we are starting a preparation of biosimilar, we have to do it in a sequential method. So step-wise procedure should be followed for the preparation of biosimilars. What we have to do? We have to check the similarities between our biosimilar with that of a reference product. We have to check the safety, efficacy and quality of our reference drug, and we have to do it in an authorized procedure to prepare a complete dossier. Dossier is nothing but a bulk of documents. We have to prepare a documentation for our biosimilar drug, and we have to compare our results with that of a reference drug. As we have discussed in the previous slide, we have to do the preparation of the biosimilar in a stepwise procedure. The two main aims is, we have to check the similarities between our biosimilar with that of a reference drug, and we have to exclude or remove the differences between our biosimilar and the reference drug. That is main aim when we are preparing a biosimilar. The first step for the initiation of biosimilar is, we have to check the analytical, quality and the characterization of our biosimilar with that of a reference drug. What is the manufacturing method that is used for the biosimilar and for the reference? What is the characters of the particular product? What is the structure of the product? What are the physical properties? What are the chemical properties? Whether the activity, biological activity of our biosimilar is similar to that of a reference compound. Whether it have same immunological properties. Whether it will, like our biosimilar may have the capability to induce antigen antibody reactions, but the reference drug may not be having. So what are the immunological properties of a test or the reference drug and our biosimilar? Whether they have any impurities in that, whether it is stable. Sometimes our biosimilar may be degraded easily, but the reference compound may be very stable. So whether it is stable or not, all this we have to compare. After the analytical and quality characterization of our test drug or a biosimilar, then second step is non-clinical studies. As you know, non-clinical. Clinical indicates which are done on the human beings, whereas non-clinical indicates which are done on the animals. It may include preclinical studies or any other studies. So in this it includes in vitro and in vivo studies. Once we are done with the non-clinical studies, then we move to the clinical studies. In this we will do the pharmacokinetic parameters, pharmacodynamic, what is the safety of that particular drug, what is the capability or efficacy of that particular drug, whether it is safe to use, whether it will develop any immunological reactions. All this can be checked in the clinical studies. So pharmacokinetics include ADME properties. Pharmacodynamics includes the mechanism, whether the biosimilar and the original compound have same mechanism, whether it is safe to use, whether the efficacy is same, whether it produces immunological reactions. All this should be compared in the clinical studies. The next step is data requirements. So we need to the proof, like in the clinical studies, we need the test in the human population. So clinical trials, as you know, we will have phase 0, phase 1, phase 2, 3, 4 and 5 post-marketing surveillance. So we need to take the participants or the human population from the local area where you are conducting a test. And in the next phase, that is in the phase 3, which can be a multi-center. That is, various countries can be included for the multi-center. Different areas are included for conducting the clinical trials. It may be in different places, so various people will be participated in the study and will get a different report. And once the clinical trials are done, the last step is post-marketing surveillance. When the product is clear from the clinical trials, it is been released into the market. After releasing into the market also, if it produces any adverse effect at any point of time, that particular drug will be banned or it will be returned from the market. Biosimilars are always compared to the innovative reference drug. We can do the biosimilar comparability with a test drug or with a standard drug or an innovative drug that is registered in our own country, or we can compare with the drug which is been registered in the other country also. Patent is something which protects your innovative idea or innovative product which you have prepared for the first time, so that no one can copy your idea or innovation. So once your patent is being done, no one can copy your innovative idea. But a biosimilar preparation can be started after 10 years of the patenting. So for example, if you have a patent for 20 years, after 10 years of your patenting, anyone can start preparing a similar drug. Not the exact drug, but a similar drug which is a biosimilar. And this preparation should be completed within 18 years. Like if you have 20 years, before 2 years of your patenting is about to finish, the biosimilar preparation also should be finished. So that in the remaining 2 years, they can have the documentation work and they can prepare the documents what are required for this regulatory review, and the particular biosimilar can be released into the market.